forensic odontology, gender determination. I'll be dividing this methods into basically three. I'll be basically dividing this into three. What are the three methods of gender determination? The three methods of gender determination are visual method or clinical method, microscopic methods, and advanced method. So gender determination or sex determination, first method, that is a clinical method. Now in this clinical method, I'll be giving you an overview of how are we going to use each and every parameter for gender determination. Like tooth size. Tooth size. See, all the teeth, if you compare between the males and females, they have the same dimensions. Except one tooth, that is mandibular canine. Mandibular canine is slightly different in males and females. Males have got a greater width of mandibular canine, compared to that of the females. And even in males, the intercanine distance is greater. So that's a very favorite MCQ that is asked, which is the tooth which is used for sexual dimorphism or gender determination. The answer should be mandibular canine. So mandibular canine exhibits a greatest dimensional difference between the males and the females. Okay, in the option they will give you mandibular canine. In the MCQ that is a framed, they'll give usually mandibular canine, maxillary canine, mandibular incisors, and maxillary incisors. Remember, mandibular canines have got the maximum dimensional difference between the males and females. Accordingly, a dental index method has been developed. And this credit goes to an Indian scientist, Rao and et al. Rao and et al. Have devised or formulated an index called as MCI, Mandibular Canine Index. Okay, so just remember here, Mandibular Canine Index was devised or proposed by. The details of this index is out of your scope. It won't be asked as an MCQ because there are a lot of complicated things in that formula that is using standard deviation, which will not be asked as an MCQ. Just what we are supposed to remember here is Mandibular Canine Index, MCI, is devised by or proposed by which person? That is the Rao and et al. Okay, moving on to the second method here, tooth morphology and sexing. What do you mean by that? How are we going to use the tooth morphology? Tooth size is over. Now what about tooth morphology? Tooth shape. How are we going to use this tooth morphology in the identification of gender? We have to remember one important ridge called as DAR. What is DAR? Distal accessory ridge. This is the ridge which is seen on the canines. Okay, it's not exactly, you can't say that it is maxillary mandibular. It's usually seen on canines, both the canines. And this is this ridge, DAR, distal accessory ridge, is very prominent on males compared to that of the females. And not only it is more prominent, frequency of this ridge being seen in males is also more compared to that of the females. So if they ask you which is the non-metric tooth morphological feature used in gender determination, and they'll give you 4 ridges, answer should be distal accessory ridge. So distal accessory ridge is the non-metric ridge that is, non-metric feature that is usually used in the gender determination. Okay, that is about certain clinical methods. Now moving on to certain orthometric methods. What do you mean by orthometric methods? We are going to use certain structures such as skull, certain bones like frontal bone, sinuses, mandible, all these things. How are these different in males and females? Generally, we have to remember that female skull, when you have got a skull, 1, 2 skull. How to identify whether a skull is a male, belongs to a male or a female? The main difference here is usually female skull has got a lighter bones, and the surfaces of these bones are also smooth. So the one important feature when you weigh, it's a crude method, but that is the way usually an investigation is done in forensic sciences. So where the first parameter would be weigh the skull. So usually they are lighter bones with a smooth surface. And if you closely examine the frontal bone, males usually have got a more lower frontal bone, and it is sloped. Frontal bone is sloped in males, whereas usually in females the frontal bone is more rounded and highly placed, higher. So that is why usually if you see in males the frontal bone is lower, whereas in females the frontal bone is higherly placed. And if you look at the shape, the frontal bone in females is more rounded and the frontal bone in males is slightly sloped. What are the other orthometric features? Zygomatic bone, all of us know. Malar bone, right? All of us know that's very commonly called as malar bone. Usually zygomatic bone or malar bone is more prominent in case of males compared to that of the females. That is why cheekbones, they say cheekbones, right? Malar bone. The cheekbones are usually more prominent in males. Moving on to the mandible, the most important, mandible. They actually say that mandible, identifying the skull, identifying the individual using mandible is a very low accuracy method. Except one important feature here is about the chin. In males, usually the chin is square shaped, whereas in females, usually it is pointed or V shaped. Okay, that is one of the crude method. However, earlier it was thought that gonial angle, what is gonial angle? Mandibular angle, the angle between the body of the mandible and the ramus, that can be used. But later it was proved that this gonial angle has got very less role in gender determination. Gonial angle has got a role in age determination. That is whether this mandible is of an old age person, whether this mandible is of a young age person. Okay, with that way we can do the age determination. So that we are going to see in the other part. Age determination, how are we going to identify? That is different part of forensic sciences. So see here for this topic today, gender determination, mandible has got very less role except for the shape of the chin. Moving on to the microscopic methods. What are the various microscopic methods that are used? One of the important microscopic method that is what was earlier used is bar bodies. All of us have heard of bar bodies. What are these bar bodies? Bar body is basically an extra inactive X chromosome. Actually, it is an X chromosome, but it is inactive X chromosome. Where do you see that? It is typically seen in the nucleus of a female cell. So bar bodies are specifically seen in females. Where do you see that? Intranuclear, within the nucleus. It is just an extra X chromosome. It appears as dark staining mass. Dark staining mass. So, suppose we get a tissue. For example, I'll get a saliva sample, or I will get a buccal mucosa smear. If I will be able to see this bar body within the nucleus, yes, I can identify that, yes, this buccal smear belongs to a female individual. Okay, so bar body is specific for females. Now the question comes, what all tissues can be used as a source of these bar bodies? We can use various tissues such as blood, saliva, buccal mucosa, buccal mucosa, and even dental pulp. Dental pulp. So dental pulp is also used for the visualization of this bar bodies. But more easily accessible way is buccal mucosa smears, where you just take the smears, smear this on a slide, stain it with a nuclear stain, acridine orange, and then look at it under the microscope. If you are able to see an extra darkly staining structure within the nucleus, that means with this that it is basically a bar body. Now, as we have bar body for females, we have something called as F bodies, and this F body is unique for that you see in case of males. This F body is typically it is unique to the Y chromosomes. F body. How to identify this F body? How to remember this? F body can be seen by a fluorescent dye. A fluorescent dye. What is that fluorescent dye called as? That fluorescent dye is known as quinacrine. So quinacrine is a fluorescent dye which binds strongly to the Y chromosome. And because of this, the F body can be easily seen under the UV light. So here the question comes, first of all, where do you see F bodies? F bodies are seen on Y chromosome. What is the fluorescent dye that is used? Quinacrine. Quinacrine. What is the source of light that is used for identification of F bodies? Ultraviolet. So similar to the bar bodies, even F bodies are also used in the identification of the gender. So F bodies are specific for males. Bar bodies are more specific for females. Moving on to the most specific method, I would say most sensitive and specific method, that is the microscopic methods or the advanced methods. Now here we are going to use certain dental DNA. All of us know we are going to use dental DNA. So DNA, all of us know, is very important has got a very important role. All of us know about this in the individual. So there are different sources of DNA. Now we have dental DNA, particularly the pulp. Dental pulp is considered to be the best source of the DNA. Okay. Now earlier, how to remove this pulp from the, how to extract this DNA from the pulp? So there was earlier one method called as a Sweet's method. Okay, Sweet was a person who devised this cryptogenic grinding method, where he used a liquid nitrogen. He cooled the tooth in the liquid nitrogen and then he pulverized the tooth. Thereby then he later extracted the DNA from the pulp. So remember from this, what is happening? You are pulverizing the tooth. What do you mean by pulverizing the tooth? you are losing the morphological features of the tooth. However, to overcome this, an Indian scientist called as Trivedi, that is popularly known as Trivedi's method, he disproved this cryptogenic grinding method, and he told a very conservative way of dental DNA extraction from the pulp. Okay, so where he actually devised a needle using which the dental pulp DNA can be taken out. So we'll not go into much details of this. Usually remember the Sweets method, they'll ask as an MCQ, cryptogenic grinding method. We have also something called as certain genetic level, certain genes which are used. One of the most important gene, that is Amelogenin or Amel gene. All of us know that human enamel has got a lot of proteins, enamel proteins, amelogenins, non-amelogenins, right? So in that, one of the important protein, that is Amel gene. This Amel gene has got a different signature. What do you mean by signature? Nucleotide sequence. All of us know about nucleotide, right? It's a basic biochemistry. Nucleotide, nucleoside. So the nucleotide size and pattern is different here in males and females. This amelogenin gene in females, remember, this gene is encoded or located on X chromosome, whereas in males this amelogenin or MLX gene is encoded on Y chromosome. So the chromosome is totally different here. In males it is located on the Y chromosome, amelogenin gene, whereas in females it is located on X chromosome. Now comes the question, how are we going to differentiate? Actually, in females where this Amel gene is present on the X chromosome, one gene has got two alleles. All of us know one gene has got two alleles, or two, we can say two genes. Allele is, a gene is like a coin. Gene is like a coin. Allele is like both the sides. One gene has got two alleles. Like one coin has got two sides. Similarly, one gene has got two alleles. In females, the amelogenin or the Amel gene has got two identical alleles. Whereas in males, the two alleles are different. The two alleles are different. Okay, so how to remember this? How to remember this becomes a bit complicated, right? How to remember this? I'll tell you simple method. What is the chromosome, sex chromosome that you see in females? XX. What is the chromosome that you see in males? XY. So similarly, remember, in females, both they are X. So similarly, the Amel gene also has got two identical alleles. Whereas in males, as it has got X and Y, so remember that way, that the amelogenin or Amel gene, two alleles are different. Two alleles are different. So if you see one gene with two different alleles, then it belongs to a male. What do you mean by different? I mean to say nucleotide sequence. So this is at the molecular level where they can identify using these genetic or genes, whether it is belongs to whether this gene or whether this DNA belongs to a male or a female individual. This Amel gene, why are we specifically talking about this gene? Because this Amel gene encodes for an amelogenin protein, and amelogenin protein is very important that you see in case of human enamel matrix. There is one more important method, that is the PCR. All of us know PCR, polymerase chain reaction. So polymerase chain reaction is also used widely in amplifying a gene called as SRY gene, that is sex determining region Y. So there's something called as SRY, also a gene that is again encoded on the Y chromosome, which is again used for gender determination. So details of this is beyond the scope, it is not usually asked. So here we are supposed to remember the names of the two important genes and how are the most important one is the Amel gene. How is it different in males? How is it different in the females? That is the question that is usually expected. Okay, so with that I come to the conclusion for today's session on the role of forensic odontology in gender determination. So in the other part of this forensic odontology, we are going to cover about the age, where we are going to use the different, about the skull, about the mandible. We are going to go into details of various methods, age determination methods in children, in adults, all those things we are going to see. And we are going to also see in the other part, there is a role of the bite marks and lip prints. That would be the second part, topics for the second part of the session of forensic odontology. I hope this part is clear, and any doubts you can please post it in the comment. I'll be very happy to solve all your doubts. Thank you.